Hello guys, welcome to my YouTube channel Regulatory Talk Service. Today our topic is toxicological screening and we will discuss about the all aspects related to toxicological screening. Let's see the introduction. As we know that toxicology is a branch of science that deals with toxic and poisons and their effects and the treatment. So therefore toxicology screening is important in toxicity for development of new drugs and extension of therapeutic potential of the existing molecule. Toxic tests are mostly used to examine a specific adverse effect or a specific endpoint such as cancer, cardiotoxicity, skin and irritation. Toxic testing also helps to calculate no observed adverse effect level and to help with the clinical study. The history of toxicity testing. The field of toxicity evolved from the 14th or 15th century from Paracelsus. As he says that all substances are poison, only dose depends on whether it is poison or a drug. A specific chemical who determine the Paracelsus determine the specific chemical responsible for toxicity of plant and animal. And second, in the field of toxicology, the contribution of Matthew Orplia is recognizable, which is a Spanish physician determined relationship between the poison and their biological properties and demonstrate the specific organ damage caused by toxin. Orplia is referred to as a father of the modern toxicology because he is involved in the toxicity poison testing from the plants and animal substance. Third is Trivan. Trivan is the next scientist which used first he used the animal in the toxicity study and he used to begin the animal in toxicity study from 1920 and to calculate the first lethal dose of animal by the W. Trivan. After that, so many guidelines and testing of toxicity testing is evolved. But first, the use of animal in the field of toxicity testing start from the Trivan. After the introduction of calculation of LD50, the next scientist is John Dress, who developed his method for eye irritation and skin irritation toxicity study, and accepted by worldwide for the testing of skin irritation and eye irritation. So we know that the DRACE test used for the eye irritation toxic testing. These are the important scientists who give their life for the toxicity studies and its development guideline. Later, after the 1920, the U.S. National Cancer Institute developed the test to identify carcinogenic chemical through the daily dosing of rats, mice for the 2 years. Through that, it develops the carcinogenicity testing guideline that is 451. In early 1960, thousands of babies were born with the debilitating birth defects caused by thalidomide, which is one of the major tragedy happens due to the lack of toxic testing of a compound. After this, all regulatory guidelines concentrated on determining toxicity profile of all pharmaceutical substance and to prepare the regulatory patient use for mandatory submission. In later 1980, the Organization of Economic Cooperation and Development, that is OECD, and International Conference on Harmonization brought about the guidelines for toxic testing. So next is the source of toxic testing. Usually the toxicants are classified based on their chemical nature, mode of action or class. The exposure class, which is classified toxicant as occurring food, air, water and soil. So the use of classified drug as a drug of abuse, therapeutic drug, agriculture, chemical, food additives, pesticide, plant toxin or phytotoxin and cosmetic. These are the sources of toxic and based on the sources, the toxicity branches divide as a environmental toxicity. Next is institute that conduct the toxic testing, that is Institute Ethics Committee under the CPCSA, that is Committee for the Purpose of Control and Supervision of Experiment on Animals. Before conducting any toxicological testing in the animal or collecting the tissue cell lines from the animal, the study should be approved or accepted by the Institutional Animal Ethics Committee. In India, the Committee for the Purpose of Control and Supervision Experimental Animal guidelines should be followed. The maintenance of experiment animal. If in your study or in your experiment there is no use of animal, then no need to take permission from this committee. An approved fluid withdrawal method should be used. Schedule Y, which is now converted into the NDCT Act after 2019, should be compiled with the fulfill of regulatory requirement. First testing method for toxicity is acute toxicity testing. In general, it is recommended that acute toxicity testing is carried out with two different animal species, one rodent and one non-rodent. In acute toxicological testing, investigational product is administered at a different single dose level and the effect of observed for 14 days. In acute toxicology testing, investigation product is administered for 14 days, so it is called as with a single administration. Observation is for 14 days. So it is used for the calculation of LD50. Determination of LD50 involves large number of animals and mortality ratio is high. Because of this limitation, the 401 is converted into the three different methods, that is the fixed dose procedure, acute toxicity category method, up and down method, which is 420, 423 and 425. If you talk about the first, that is fixed dose procedure, is used to assess non-lethal toxicity rather than the lethal toxicity because the end point of fixed dose procedure is different as compared to acute toxicity craft method and up and down method. The investigational product is administered at a fixed dose, that is 5, 50, 400 and 2000, and the experimental animal is observed for the specific period. Next method is acute toxic category. Acute toxic category method is a sequential procedure in which three animals from the same sex is used for the each step. For any acute toxic testing, it is recommended that only one sex or same sex is used for the completion of the study. In acute toxic class category, the screening method for pre-identified starting doses may be used, and the test dose should be selected based on the globally harmonized statistics GHS classification system. Next is up and down method. The up and down testing approach is also known as a staircase design because in that the single dose is fixed. After that, the upper dose and lower dose is selected based upon the result. So, in this toxicity testing, the approach most recommended by the various regulatory agencies because the method reduces number of vertebrate animals in the research. Female rodents are favorable for the up and down testing, and up and down testing is limited for the dose of up to the 2000. In the above method, that is fixed dose procedure and toxic category, we can use the dose up to 5000, but in up and down, 2000 mg per kg dose is fixed. Acute toxicity for inhalation. Same as oral dosing, acute inhalation toxicity testing is performed for aerosol-like preparations, which is given by inhalational route. And the species which is rat is most preferable animal for inhalational toxicity study. For any toxicological study, the animals are acclimatized before the experimentation. So they are maintained in the air of flow, that is 15 to 12 to 15 air changes per hour with adequate oxygen, that is 19% per. So this condition is only for the acute inhalational toxicity, not for all toxicity. So it is necessary to maintain the 12 to 15 air changes per hour. Animal is exposed to the test substance for minimum 4 hours and then it is monitored for 14 days. Same as in acute oral toxicity, administration is for only single administration. Because of it is an inhalational toxicity, exposure is for 4 hours and then it is monitored for 14 days. During the observational period, animal is observed for the tremor, convulsion, salivation, diarrhea, lethargy, sleep, and coma. Dead animals are examined for the histological and pathological changes. At the end of the study, animals are sacrificed and pathological changes are evaluated as same in the acute toxicity testing conducted by orally. Next is acute toxicity testing for topical preparation. The eye irritation test and skin irritation test provided by the DRES formula is very important for topical preparation. Dermal and ophthalmic preparation can be tested by using the DRACE test. So it is worldwide accepted that irritancy, that is skin irritation and eye irritation, by the DRACE test. That is DRACE eye irritation test and DRACE skin irritancy test are used to measure harmful effect of a chemical on the rabbit and guinea pig. Most preferred species is rabbit for the skin irritancy test and guinea pig both. In eye irritancy test, 0.5 ml of test substance is administered to an animal eye and the animal is restrained for 4 hours. Redness, swelling and discharge alteration, hemorrhage and change in opacity, blindness are assessed for 14 days after the 0.5 ml of test substance administration into the eye. Same as the skin irritation, the substance that is 0.5 gram substance test substance is applied on the surface of animal skin. During the observation of 14 period, same sign such as erythema, edema, redness, these things are observed uh, in the area, that is topical area, applied area. Some alteration in in vitro testing are methods available for the uh, acute toxic testing of topical preparation instead of the use of animal. Uh, this alternative in vitro testing is observed, and the given guideline is like 439. Next is skin sensitization test. The difference between skin irritation and skin sensitization is to only that without irritation only if any substance or the drug which activate the immune system, that is uh, skin sensitization effect, and this effect is assessed by the DRACE test, open epicutaneous test, optimization test, split adjoint test, GPMT, that is one year peak maximization test. Main important is local lymph node assay, LNNA. The LNNA method is used as an alternative to guinea pig DRACE test, and it is widely accepted. This method meets regulatory requirement also. In LNNA test substance is applied on the surface of ears of a mouse for the 3 consecutive days, and proliferation of lymphocyte in the draining lymph, that is ear lymph node, is measured at the end. Next is the mutagenicity testing. Mutagenicity testing is used to assess the submicroscopic changes in the base sequence of DNA or the chromosomal abbreviation and structural abbreviation in the DNA, including duplication, insertion, inversion, and translocation. In vitro testing is carried out in a 2 or 3 different bacteria and mammalian cells also to cover the endpoints of gene mutation, clastogenicity, and aneuploidy. The test generally includes bacterial reverse mutation assay, that is AIMS assay. The choice of an additional test depends on the chemical structure. In vivo mutagenicity test, which is a dose dependent and is used to determine case by case basis. Mutagenicity study with the transgenic animals are more appropriate assay techniques to determine the toxicity of a test substance. So the next is a carcinogenicity testing. Both rodents and non-rodents are same in acute toxic testing. In carcinogenicity testing also used. The tests are carried out over the greater portion of animal lifespan, that is 2 years. If they are not found, the test may be terminated after the 18 months in the case of mice or 14 months in the case of rats. If the animals are healthy, hematological analysis is performed after the 12 months and 18 months respectively. Sometimes it is performed within the 6 months also. The animals are sacrificed and gross pathological changes are noted, and histologic study carried out on the basis of tissue. Next is one generation reproduction toxicity. The test compound is administered in this test for toxic testing both in male and female. Administration is for duration of 1 complete spermatogenesis cycle in the male or 2 complete estrous cycle for a female animal. Rodents are prepared for the one generation reproduction toxic testing, which is a rat. The test compound is administered to the female during the period of pregnancy and nursing. The sperm of male animals are collected and the sperm morphology and mortality is analyzed. In this study, the basic things is the administration of a compound and duration of administration, which is in the male, if you talk about male, one complete spermatogenic cycle and two complete estrous cycle. Estrous cycle is 4 days, like menstrual cycle in the human. Same in animal, estrous cycle is. If we talk about 4 days for one, 4 to 5 days for one estrous cycle, then for two estrous cycle, that is minimum 10 days administration of the compound, and the test compound is administered in the female. Parturition, the number of offspring and their sexes are recorded, and the number of dead and live pups are recorded. The weight of live pups and in the morning and evening each day during the first 4 days is recorded. After the termination of study, animals and pups are sacrificed and subjected to the histological examination for the toxic test. Next is two generation reproductive toxicity studies. Same the procedure followed in one generation toxicity study, we also follow in the two generation toxicity study. If we consider one generation toxicity as a F1, then after the parturition and nursing rat, administer the test drug and mortality of pups is observed. From F1 generation, one male and one female animals is selected. That is from the pups, one male and one male and one female is selected. The same procedure is repeated to get F2 generation offspring, and F1 offspring is not allowed to mate until they have attained full sexual maturity, and pair without a pregnancy are evaluated for the infertility testing for one generation toxicity, and next also of histological examination carried out at the end of the study. The pups and all animals are sacrificed, gross pathological and histological examination carried out on the animal. Next is the toxic kinetic study. Without toxic kinetic study, we are not able to calculate no observed adverse effect level. Toxic kinetic, which is the extension of pharmacokinetic, deals with the kinetic parameter, higher doses of chemical and toxic xenobiotic. Toxicokinetics help the study metabolism, excretion pattern of xenobiotic. Animal toxic kinetic data helps to explore the physiological base of physicokinetics and humans. In toxicological testing, pharmacokinetic studies are usually carried out in the rodents, rabbits, dogs, non-human primates, and swine using the many routes of administration. Blood samples are collected at various time points to analyze pharmacokinetic data such as area under curve, drug distribution ratio, Cmax, Tmax, or other pharmacokinetic parameters. Same in the toxicokinetic study, this blood collection is carried out. Toxicokinetic study may be performed using in vitro cell lines also, but the most preferred method for toxicokinetic is in vivo toxicokinetic study. Next is neurotoxicity study in the rodents. The effect of taste substance on the central nervous system can be studied through the neurotoxicity study. Neurotoxicity study may be employed to evaluate specific histopathological and behavioral neurotoxicity. Usually, neurotoxicological studies are carried out in the adult rodent. The test substance may be administered for 28 days or even more than 90 days. Neurological changes are evaluated by following the neurotoxicological test. In 1998, the in vitro model for neurotoxicity was developed, and various regulatory agents now recommended for in vitro neurotoxicity test. Developmental toxicity and embryo toxicity study. Embryo toxicity study can be studied using both in vivo and in vitro methods. Rodents are preferred in vivo toxicity. Compound is administered between 8 to 14 days of pregnancy, and embryo lethal effects are studied. At the end of the study or on even 21st day of study, the cesarean section is performed, and parameters such as fetus with hemorrhagic bullae, limb malformations, cleft palates, open eyelids, tail deformity, as well as mortality and number of deaths and live pups are noted. Embryo toxicity study can be performed using in vitro methods such as embryonic stem cell test for embryo toxicity, micromass embryo toxicity assay, whole rat embryo toxicity assay. Next is the genetic toxicity testing. Genetic toxicity testing are used to identify gene mutation, chromosome changes, alteration in DNA sequencing. These tests are usually conducted in various species, including whole animal, plant microorganism, and mammalian cells. In the whole animal model, rodents are preferred Genetic toxicity assessed using rodent chromosome assay. Dominant lethal assay, mouse-specific locus assay, micronucleus test, heritable translocation assay, and sister chromatid exchanges. Next is a repeated dose toxic testing. This test is used to for the repeated administration of a drug by oral route, inhalation route, topical route. So repeated dose toxic testing is carried out for minimum 28 days. The test substance is administered daily for certain period through the oral route. If the route is not convenient, the test substance may be administered parenterally. The test substance is administered regularly at the specific time, usually the rodents of any gender, age of 5 to 6 weeks, or other age is 7 to 8 weeks. Repeated dose toxic testing. This should be the little individual variation between the animals, and allowable variation in the body weight is plus or minus 20%. For randomization, we need to check whether it is within the plus or minus 20% or not. Satellite group may be included in the study protocol. This gives the idea about delayed toxicity or recovery, if any toxicity of high dose. The basic parameters such as behavioral biochemical parameters of the animal should be recorded. Major difference between the repeated dose and subchronic toxicity study is the duration. That is 28 days per subject or 90 days for subchronic. Next is the chronic oral toxicity testing. Chronic toxicity studies are conducted within the minimum one rodent and one non-rodent species. The same in acute toxicity for within one rodent or one non-rodent. The test compound is administered over more than 90 days. Animals are observed periodically. The chronic toxicological study provides inferences about the long-term effect of test substance in the animal and may be extrapolated to the human safety of the test substance. There should be little individual variation between the animals and allowable weight variation, that is 20 plus or minus 20% for same as subacute toxicity, subchronic toxicity, and chronic toxicity. In these also satellite groups are used, but the number of animals in satellite group is increased, which is double, that is a 10 number per sex. During the study period, the animals are observed for normal physiological function, behavioral variations, and alteration in the biochemical parameters. Thank you so much. These are the different toxic testing which we are observed for the toxicological screening.